Hello students, we're back, and DNA is a double helix. That's the structure of DNA. And when cells divide, which is really the process in which they're making more cells, so it's really multiplication, but we call it cell division. DNA has to also be copied. So if we look at DNA, this is just a picture of the structure of DNA, right, on its side. Now, when DNA replicates, it has to make copies of itself. So then, when a cell divides, it's called cell division, 2 new cells are made. All right, this cell has DNA in it. These cells need DNA also. And when a cell does this, it's copying itself. So these cells are the same as this one. And these are called the daughter cells. I don't know why they're not called sons, daughters. And this is the parent cell, or the original. These are identical, and they're identical to this. So they have the same DNA, but to get the same DNA, they have to copy the original. So let's say this is the DNA in this cell. Okay, we have to make 2 of them because we're going to divide. So to do that, there's a process called DNA replication. And that's just what it says it is. It's DNA making copies of itself. So DNA copying. And there's a lot of steps to this process, but for this class, just know that we're making 2 DNAs from 1 DNA. And to do that, the DNA will unwind. All right, so first it has to be untwisted. And there's enzymes like helicase, like a helix, helicase, untwist the DNA. That's an enzyme. An enzyme is a chemical that speeds up a reaction. That's what enzymes do. And enzymes are not changed in a reaction, they just speed it up. So they speed up reactions, and a lot of enzymes end in ASE. So prefix or suffix, ASE, suffix is at the end of the word. Enzyme. It's an indication of an enzyme. OSE is indication of a sugar, such as deoxyribose. That's the sugar found in DNA. ASE, enzyme. So if you think of the DNA structure as a double helix, the enzyme that untwists it, straightens it out, is helicase. Helix, helicase, you just put the ASE on it. All right, and there's DNA polymerase, there's RNA polymerase, there's lots of enzymes in this process. You don't have to know the enzymes' names and the functions. You do have to know what DNA replication is. So we're copying DNA. So when the first thing, I'm going to untwist it. Then we're going to separate the strands apart, so we can make copies. And then, remember, these are bases, right? So we untwist, separate the strands. Now I'm going to just make up the sequence here. I'm just going to say this is T, C, G, A, G, C, A. Okay, so if this is the sequence A, T, C, G, A, G, C, A, the complementary strand then is going to be T, because A, T, and T go together. A, G, C, T, C, G, T. All right, so this side then is going to make DNA, and this side is going to make DNA. So we have a new strand comes along was added to both sides. And in the end, you're going to get 2 double helix structures. So 2 DNAs are going to be made. So this is the process of DNA replication. And this is important for cells to divide. When cells divide, they make more cells. And to do that, they have to divide, they have to make more DNA. So this is in the nucleus of the cells. So nucleus, in the nucleus, DNA replication. Because think about it, DNA is very valuable, right? Contains information, makes you who you are. There's a lot of studies being done on how much of you is actually programmed by the DNA and how much is due to your environment. So we're talking about nature versus nurture. Nature being the DNA, nurture your environment. You're not all just responding to what your DNA tells you, but DNA is really instruction manual for life. And finding out more and more things are actually genetic than we originally thought. Obviously we know they're genetic diseases, but we're also learning some cancers can be, people, if they have certain genetic codes, that they can be more predisposed to developing cancers, mental illnesses, personality. These are things we're studying now, and we're learning that there is a lot of information in the DNA. Okay? But this is DNA replication, so DNA copying itself. If we have an instruction manual, let's say you, or think of Tesla. The engineers that designed Tesla are not the ones actually building the cars. The engineers are separate from the factories. Just like in your body, the engineers then, the ones that actually design the Teslas, let's say, are like the DNA. It's instruction manual. DNA is instruction manual for life. Let's say the engineers create designs for building cars, or the designs, that's the plan. But the cars are built in a separate place, in a factory, right? Just like in your body. So we have information in the DNA, in the form of the DNA, but we got to get that information to make things for the cell, such as proteins. So really, our body's made up of proteins. And proteins are made of carbon, hydrogen, oxygen, nitrogen. Those are the main elements. There's other elements involved, but really our body is mainly proteins. That's what we see. So we have to get the instructions, which is in the DNA, to the factory where the proteins are made. So in our body we're making proteins, in a car factory we're making cars, Teslas. So in our body, in our cells, the factory is outside of the nucleus. So just like a car factory is separate from where the engineers are designing it. So the factories to make proteins are in ribosomes in the cell. So the ribosomes, remember, where proteins are made. Proteins. And the ribosomes are outside of the nucleus. So we got to get the information from the DNA, which is in the nucleus, to outside of the nucleus in the ribosomes. And how do we do that? We can't take the DNA and move it out because it's way too valuable. It's risky to damage it. Because when we get outside the nucleus, there's no protection anymore. It's like walking outside where people aren't following the social distancing guidelines. It's dangerous out there. Dangerous outside of that nucleus. Alright? So the DNA is in your cell is in the nucleus. There's also DNA in the mitochondria that I talked about before. We're looking at the nuclear DNA. We have to get that information though from the nucleus to the ribosomes. And how do we do that? We can't move the DNA out. So we have to have a messenger. We There's got to be something that takes the message from DNA to the ribosomes to make proteins. We call that RNA.